A cockroach is sitting on a surface doing nothing in particular. A small metallic green and fully formed and immediately ready to begin hunting wasp lands directly beside it on the same surface. The cockroach does not run away from the wasp landing next to it. It does not attempt to escape in any direction or by any means available to it. It simply stands there while the wasp stings it twice in very specific locations in its body. Then the cockroach begins grooming itself slowly and repeatedly in the dark of the sealed burrow, itself slowly and methodically, cleaning its antennae and body surfaces in a slow, compulsive repeating pattern, while the wasp walks away briefly to prepare the next stage of the process. When the wasp returns, she takes the cockroach by its antenna and walks it, on foot, directly into a small burrow prepared in the ground nearby. The cockroach follows through the antenna, grip the entire distance to the burrow entrance, without pulling away from it or resisting the direction of travel in any way, into the burrow, without any resistance whatsoever, as if this is a voluntary walk with a companion rather than anything threatening. Inside the burrow, the wasp lays a single egg on the cockroach's leg. She seals the burrow entrance with pebbles, plant material, and small stones, and leaves the cockroach sealed inside alive with the egg attached to its leg. The cockroach remains in the burrow alive and grooming itself slowly and repeatedly in the dark of the sealed burrow itself. The cockroach will remain in that burrow alive for approximately the next 4 weeks. The wasp larva hatches, feeds on the cockroach from the outside, then from the inside. And eventually, the cockroach dies when there is nothing left to sustain it. The wasp that emerges is a new adult ready to repeat the entire process. Today we are talking about Ampulex compressa, the emerald jewel wasp, an insect that does not kill its prey in the way most predators kill. It does something that is considerably more disturbing than straightforward killing. It removes specifically the cockroach's ability to want to escape on its own initiative, and leaves the cockroach sealed inside alive with the egg attached to its leg, everything else intact. Let's start with what this animal actually is before we get into the neuroscience. The jewel wasp belongs to the family Ampulicidae and is a completely solitary wasp species. It is found across tropical regions of South Asia, Southeast Asia, the Pacific Islands, and parts of Africa. Individual females are approximately 2 to 3 centimeters long in body length. The female is metallic emerald green with reddish-orange legs. And visually it is genuinely striking, the kind of insect that if you saw it on a wall, you would stop and look. It does not build communal nests or live in colonies the way honeybees or paper wasps do. Each female hunts provisions and reproduces entirely independently from other individuals. She mates, finds prey, provisions a burrow, and reproduces entirely alone. And the prey she requires for reproduction is specifically the American cockroach, Periplaneta americana, one of the largest common cockroach species, and one that is significantly larger than the wasp herself. The cockroach can weigh 3 to 4 times what the wasp weighs. It has legs capable of carrying it at significant speed when threatened. It has antennae that detect air movement and trigger escape responses in milliseconds. Under normal circumstances, a cockroach that detects a threat turns and runs before most predators can close the distance. The jewel wasp cannot outrun a cockroach that has already begun its escape response. And a cockroach that fights back can seriously injure or kill a wasp this small. So the wasp does not attempt to chase or physically fight a fully mobile cockroach. She removes the chase and the fight from the equation entirely, before a chase or fight becomes necessary, by removing the cockroach's ability to initiate either. The first sting goes into the cockroach's thorax, the middle section of the body between head and abdomen. This sting delivers venom that temporarily paralyzes the cockroach's front legs. The paralysis lasts only a few minutes, just long enough for the wasp to position herself correctly for the second sting. The second sting is the one that produces the remarkable behavioral effect. It is delivered directly into a specific region inside the cockroach's head, specifically into a region of the cockroach's brain called the subesophageal ganglion. This structure is responsible for initiating and sustaining the escape locomotion response. It is the part of the cockroach's nervous system that says run when something threatening appears. The venom delivered by the second sting does not kill neurons in this region. This sting does not damage or destroy the cockroach's brain tissue in any gross anatomical sense. It blocks a specific neurotransmitter receptor I, the octopamine receptor. Octopamine functions in invertebrate nervous systems the way adrenaline functions in vertebrates. It is the compound that drives the cockroach's fight-or-flight response. When the octopamine receptors in the subesophageal ganglion are blocked, after the sting, the cockroach retains complete motor function in all its limbs. It can still walk, it can still groom, it can still respond to many stimuli. But it cannot initiate the spontaneous escape locomotion that would normally carry it away from danger. Researchers studying this effect describe it as a specific and targeted suppression of escape motivation. It is not a general sedation or paralysis of the kind most venomous animals produce. If you pick up a jewel wasp-stung cockroach and place it in a threatening context, it will often walk somewhat normally in response to being moved. But it will not spontaneously generate its own escape behavior without a direct external stimulus. Placed back on a neutral surface, it returns immediately to the slow, compulsive grooming pattern. The wasp has not made the cockroach physically unable to move or walk. She has removed the cockroach's ability to generate the motivation to move on its own initiative. This specific distinction between unable and unwilling matters enormously for the wasp's reproductive strategy. A paralyzed cockroach would deteriorate rapidly as a food source. A dead cockroach begins decomposing immediately and deteriorates even faster than a paralyzed one. She needs the cockroach to remain a fully living functional organism for approximately 4 weeks, while her larva completes its full development inside that enclosed space. Dead prey begins decomposing immediately and cannot serve as a viable food source for that duration. Only living prey maintained in a specific controlled behavioral state can serve this function. And that is precisely what the second sting creates, a living cockroach held in behavioral suspension. Now let's talk about the grooming behavior specifically, because it is one of the more striking visible effects. After the second sting, the cockroach begins an extended grooming episode, cleaning its antennae, legs, and body surfaces with its mouth parts in a slow, repetitive pattern. This grooming episode lasts approximately 30 minutes before the cockroach settles into its passive state. Researchers believe this extended grooming serves a specific functional purpose for the wasp. It keeps the cockroach stationary and occupied in a predictable location, while the wasp finishes preparing the burrow and makes her return trip back to collect it. It also ensures the cockroach is clean of external debris or organisms that might compete with or harm the wasp larva developing on and eventually inside it. The grooming itself, slowly and repeatedly in the dark of the sealed burrow, is not random behavior that happens to follow the sting. It is a specific behavioral program that the venom appears to trigger deliberately. The venom contains multiple compounds beyond the octopamine blocker, and researchers are still working to identify which components produce which specific behavioral effects. The full pharmacological profile of jewel wasp venom is not yet completely characterized, which is itself a significant statement given how extensively this insect has been studied. Now let's address what happens next, specifically what occurs when the wasp comes back. She does not drag the cockroach the way most parasitoids drag paralyzed prey to their burrow. She does not carry it or drag it in the way most wasps transport paralyzed prey. She leads it. And this distinction is what makes the jewel wasp unusual among parasitoids. She takes hold of one of the cockroach's antennae, not biting it off, just grasping it, and simply walks toward the prepared burrow with the cockroach following behind. The cockroach follows through the antenna, gripped the entire distance to the burrow entrance without pulling away or resisting. This is the detail that researchers emphasize as the most behaviorally remarkable aspect of the whole process. A cockroach being led by an antenna into a hole in the ground, following a wasp into an underground chamber on its own 4 legs. It follows without resistance, without escape attempts, and without any visible distress response and without visible distress. Without any escape attempts, without any resistance, and without visible distress, showing no behavioral signs that anything threatening is happening to it. The cockroach is not restrained in any meaningful physical sense during this walk. If it wanted to pull its antenna free from the wasp's grip, it has the strength to do so. If it wanted to turn and run, it has the motor capability to do so. It simply does not generate the internal motivation to initiate that behavior on its own. Inside the burrow, the wasp lays a single white egg on the underside of one of the cockroach's front legs. The egg is glued there with a secretion from the wasp's abdomen. It will hatch in approximately 2 to 3 days after being laid. During those 2 to 3 days, the cockroach remains in the sealed burrow in its passive behavioral state. It does not attempt to leave the burrow or move toward the sealed exit. Researchers have confirmed this by marking stung cockroaches and checking their position periodically. In every documented case, they stay inside the burrow without any attempt at escape. The wasp seals the burrow entrance with debris, pebbles, plant material, small sticks. This seal keeps the cockroach inside and reduces access for potential competitors or parasites. When the larva hatches, it immediately begins feeding on the cockroach's surface, consuming hemolymph, the insect's circulatory fluid, through a precisely chewed opening in the cuticle. The feeding pattern the larva follows is structured and sequential in a consistent way. The larva feeds from the outside of the cockroach for several days. Then after several days, it chews its way through the cuticle to feed from inside. And once inside, it continues feeding on the cockroach's internal organs in a specific sequence, deliberately leaving the most critical life-sustaining organs to be consumed last. Specifically, the heart and major ganglia, the nerve centers that maintain basic body functions, are consumed only in the very last stages before the larva is ready to pupate. This sequencing keeps the cockroach alive and therefore the food fresh for the entire developmental period. Researchers analyzing the sequence have found it is too consistent across many observations to be random. The larva follows a feeding sequence that maximizes how long the cockroach remains viable as food. This behavioral program in the larva is instinctive. The larva has never learned this sequence. It executes this sequence correctly from the very first feeding on its very first host. Approximately 4 weeks after the initial sting, a fully developed adult jewel wasp chews through the cockroach's cuticle and emerges into the burrow, and then chews through the sealed burrow entrance into the open air. The cockroach that was sitting alive in the burrow 4 weeks ago is now completely consumed. Now let's talk about what this system reveals about neuroscience specifically, because the jewel wasp's manipulation of the cockroach's behavior has become a significant research tool for understanding how escape motivation is controlled in invertebrate nervous systems. Researchers working on this question have found that the jewel wasp's venom provides a remarkably precise intervention in one specific neural circuit rather than across the brain broadly. Most chemical interventions in neuroscience research, whether drugs or genetic manipulations, have broad effects, affecting multiple systems and making it difficult to isolate the contribution of any single circuit or receptor type. The jewel wasp's venom acts on a very specific target in a way that produces a very specific behavioral change, without dramatically altering other behavioral systems or motor functions simultaneously. This makes it a useful tool for asking specific questions about how escape initiation works at the circuit level. Researchers have used jewel wasp venom to probe the role of octopamine signaling in locomotion control, and the results have contributed to broader understanding of how neuromodulators regulate behavioral states, not just in cockroaches but as a model for understanding similar regulatory systems in other animals. The subesophageal ganglion manipulation also raises questions about what consciousness and volition mean at the neural level. A cockroach that can walk but does not choose to walk on its own. A creature that retains motor capacity but has had specific motivational capacity removed. Sits in an uncomfortable conceptual space between normal behavior and complete incapacitation. It is neither fully functional in a normal behavioral sense nor incapacitated in a physical sense. It is specifically modified in one narrow behavioral dimension, and this precision is what makes the jewel wasp's intervention so interesting to researchers, beyond what it reveals about this specific insect's behavior. Now let's talk about where jewel wasps fit in the broader category of parasitoid wasps, because the jewel wasp is not alone in using a living host for larval development. Parasitoid wasps are one of the most species-rich groups of insects on Earth. Estimates suggest there may be 100,000 or more parasitoid wasp species in total, making them collectively one of the largest animal groups by species count. They use an enormous diversity of host animals, caterpillars, beetles, flies, spiders, and many others. And they have evolved an equally enormous diversity of manipulation strategies. Some parasitoid wasps inject venom that mummifies the host while turning it into a preserved but living food source. Some inject compounds that alter the host's behavior to protect the developing larvae from predators. The host caterpillar of certain Glyptopanthes wasps actually defends the cocoons of the wasp larvae, attached to the outside of its own body and feeding on what is left of it, thrashing its head at approaching predators as if the cocoons were something worth protecting. The caterpillar whose innards fed those larvae is now actively guarding the parasites that consumed it from external predators and parasites that might attack the wasp larvae attached to it. The jewel wasp is remarkable among parasitoids for the sophistication and precision of its manipulation. The two-stage sting process with anatomically targeted delivery is more precise than most parasitoid venom systems. The behavioral state it creates, ambulatory but non-initiating, is more functionally specific than simple paralysis or mummification achieved by most other parasitoid species. And the leading by antenna behavior that the venom enables is genuinely unusual, even within this diverse group. Most parasitoids simply paralyze their prey completely and drag the immobile body to a prepared location. The jewel wasp has the cockroach walk itself to its own imprisonment. Now let's talk about the research methods used to understand this process, because studying the jewel wasp has required some creative experimental approaches. Early researchers studying the sting behavior could observe the gross behavioral effects easily. A cockroach that does not run after being stung is straightforwardly observable. But understanding which specific brain region was targeted required neuroanatomical work. Identifying the subesophageal ganglion as the target took careful comparison of sting injection sites with the behavioral effects produced by venom injected into different brain regions experimentally. Researchers injected venom directly into different cockroach brain regions and recorded which injections produced the characteristic non-escape behavior. The subesophageal ganglion injection reproduced the effect most precisely, which then allowed researchers to identify the octopamine receptor as the likely target. By testing whether blocking octopamine receptors pharmacologically produced similar behavioral effects, it did. The pharmacological block produced an identical behavioral effect to the natural sting. This convergence of approaches, behavioral observation, anatomical analysis, and pharmacological testing, established the mechanism with a degree of confidence that any single approach alone could not provide. Subsequent work characterized the venom's composition more specifically, identifying multiple peptide components beyond the octopamine targeting compound, some of which appear to contribute to the extended grooming itself, slowly and repeatedly in the dark of the sealed burrow response, and some of which may play roles in the preservation of host tissue quality during larval development. This research continues, and the full venom pharmacology remains an area of active investigation, meaning a 3-centimeter emerald wasp that hunts cockroaches for a living, is still generating research publications in neuroscience and biochemistry, more than 3 decades after the basic behavioral phenomenon was first described systematically. Write in the comments if you knew about the jewel wasp before today, because I think the reaction most people have when they first hear this described is that it sounds like something from a science fiction story rather than something that happens in backyards and vacant lots across tropical Asia and Africa right now. A wasp approximately the length of a human finger and fraction of its weight, giving a cockroach a targeted injection in a specific brain structure, and then leading that same cockroach home by its antenna into an underground chamber. Because the second sting left it with nowhere it wants to be and nothing it wants to do, there is a specific detail about the jewel wasp's sting precision that became clear only when researchers began imaging the injection process directly. The second sting does not penetrate the cockroach's head randomly. The wasp probes the surface of the cockroach's head with the tip of her stinger. Feeling for specific anatomical landmarks before committing to the injection, she uses mechanosensory feedback through the stinger itself to locate the correct entry point and to confirm she has reached the target brain region during the injection. This was demonstrated experimentally by studying wasps that were given cockroaches, with their subesophageal ganglion artificially displaced slightly from its normal position. The wasps spent significantly longer probing before stinging, and their stings were more likely to miss the target when the ganglion position was altered. This implies the wasp is performing a form of real-time tactile navigation inside the cockroach's head, using the stinger as a sensory probe before committing to venom injection. This level of precision in a 3-centimeter insect navigating inside the skull of its prey, using real-time tactile feedback transmitted through the stinger itself like a needle probe, remains one of the more remarkable behavioral examples documented in entomology. It is precisely the kind of finding that causes researchers who have spent years studying other insects to sit back and reconsider their assumptions about what small nervous systems are capable of. Now let's talk about what the jewel wasp's behavior tells us about the evolution of complex behavioral programs. The entire sequence, locate cockroach approach, deliver first sting, wait, deliver second sting with precision, trigger grooming wait, return lead by antenna provision, burrow lay egg seal exit, is genetically encoded. No jewel wasp learns any of this behavior from a parent or from observation. Each wasp performs the complete sequence correctly on its first attempt, with no prior experience with cockroaches or burrows of any kind. This is instinctive behavior in the most precise and literal meaning of that term. A completely genetically determined behavioral program that executes without learning or practice. And that program includes the precision targeting of a specific brain region. In a prey animal the wasp has never personally encountered before in any context. Researchers studying the evolution of this kind of complex instinctive behavior face a genuinely challenging and still unresolved theoretical question about evolutionary pathway. How does a behavior this precisely adapted to a specific target evolve incrementally? Each step in the evolution of the jewel wasp's hunting sequence presumably had to provide some selective advantage in the intermediate forms, before the complete two-sting system and leading behavior were fully in place. A sting that partially suppresses escape motivation would be more useful than no suppression. A partial suppression that allows even some leading behavior to work would be more useful than complete paralysis. Researchers believe the full sequence evolved gradually from simpler ancestral forms, in which cockroaches were simply paralyzed and dragged rather than led, with the two-sting sequence replacing a single generalized paralysis sting progressively, as behavioral precision became more advantageous than brute paralysis in the competitive environment. This kind of stepwise refinement of a complex behavior is difficult to study directly, because the intermediate forms no longer exist to be examined. But comparative analysis of related wasp species that use simpler cockroach manipulation strategies provides partial support for this evolutionary reconstruction of the sequence's origins. Now let's talk about the cockroach that the jewel wasp targets in more detail, because the American cockroach that the jewel wasp specifically targets is itself an extraordinary insect that does not get adequate attention given its capabilities. Periplaneta americana is one of the fastest terrestrial insects known. It can run at approximately 1.5 meters per second, which represents roughly 50 body lengths per second. For a human to run at equivalent speed relative to body length, they would need to run approximately 300 kilometers per hour. The cockroach's escape response begins in the cerci, sensory organs at the rear of the abdomen, that detect the air movement patterns generated by an approaching predator. The signal travels from the cerci to the leg muscles via giant interneurons, some of the largest and fastest conducting neurons found anywhere in the insect world. From stimulus detection to the first leg movement takes approximately 50 to 100 milliseconds. This response speed is sufficient to begin escape before most predators can complete a strike. The jewel wasp cannot beat this response time in a direct approach, which is why she has to eliminate the response entirely through chemical intervention, rather than simply moving faster than the cockroach's escape initiation. The giant interneurons that enable this rapid escape response are specifically in the circuitry that the jewel wasp's second sting targets. By blocking octopamine signaling in the subesophageal ganglion, the venom interrupts the signal that would normally activate these fast response escape circuits. The cockroach retains the physical capacity to run at 1.5 meters per second. It simply never receives the internal neurochemical signal that it should initiate that sequence. This is the specificity that makes the jewel wasp's venom so interesting to neurobiologists. It does not slow the cockroach's muscles or reduce its physical running capability. It does not weaken the cockroach structurally or drain its energy reserves. It specifically removes the neurochemical switch that initiates the escape sequence. while leaving the entire escape machinery intact and ready to run if only it received the order. Now let's talk about what happens in the burrow during the 4 weeks of larval development in more detail, because this is where the biology becomes uncomfortable to think about in specific terms. During the 2 to 3 days between egg laying and hatching, the cockroach remains in the sealed burrow in its passive behavioral state. It grooms, occasionally shifts position slightly, and remains metabolically alive. Researchers who have opened burrows during this period describe cockroaches that are unremarkable looking, sitting in a small chamber showing no signs of distress or any attempt to escape their situation. When the larva hatches, it attaches to the surface of the cockroach's thorax near the leg and chews a precisely located small feeding hole through the cockroach's outer cuticle. This first feeding hole does not cause the cockroach to struggle significantly. The cockroach's pain and distress responses appear to be sufficiently suppressed, or the initial wound is sufficiently small that the distress threshold is not crossed, that no dramatic behavioral change occurs when feeding begins. The larva feeds continuously from this exterior position for several days, growing visibly larger across the body as the external feeding period continues. Then it chews its way inside and begins the internal feeding stage. The cockroach continues to be alive throughout the early stages of internal feeding. Basic cardiovascular function and major nervous system activity continue throughout. The cockroach may move occasionally or respond minimally to disturbance during this period. It is not fully dead until the larva has consumed enough internal tissue that the cockroach's basic life-sustaining processes can no longer function. Researchers have found that the precise consumption sequence, peripheral before central, fatty tissue before neural tissue before major ganglia, extends cockroach survival by an estimated several days compared to random feeding that would damage critical systems earlier. This sequencing appears to be genetically programmed in the larva, and it is specifically adapted to the American cockroach's anatomy. The jewel wasp larvae that are given different cockroach species show less efficient feeding sequences, suggesting the specific consumption program is tuned to the normal prey species, rather than being generalized across all possible cockroach host species equally. Now let's address a question that sometimes comes up in discussions of the jewel wasp, specifically whether what happens to the cockroach constitutes suffering in any meaningful sense. This is a question that is genuinely difficult to answer with current scientific tools. Cockroaches have nociceptors, sensory neurons that respond to damaging stimuli, and they show behavioral responses to damage under normal conditions. Whether these responses involve subjective experience in any sense analogous to human suffering is not something current neuroscience can establish with any confidence. The cockroach's reduced behavioral state following the jewel wasp sting suppresses some of the behavioral expressions of distress response, but whether it also suppresses whatever subjective experience might or might not underlie those behaviors is completely unknown and likely unknowable with any current research method. Researchers who work on insect cognition and pain perception generally approach this question carefully, acknowledging significant uncertainty about what insect subjective experience involves, if anything. The jewel wasp's venom targets a specific behavioral output, escape motivation. Whether it also affects whatever internal experience a cockroach has is a question current biology cannot answer and may not be able to answer for a very long time. This uncertainty is worth acknowledging directly rather than defaulting to either easy answer. Because the easy answer in either direction, cockroaches definitely suffer or cockroaches definitely don't, would both require a level of confidence about insect subjective experience that we do not have. Now let's talk about what the jewel wasp represents within the broader story of parasitoid insects. Because parasitoid behavior is one of the most species-rich and ecologically important phenomena in the entire insect world and among the most species-rich phenomena in all of biology, the ichneumon wasps, a family of parasitoids that Darwin famously cited as evidence against a benevolent creator, are estimated to comprise between 25,000 and 100,000 species on their own as a family, making them potentially the most species-rich family of animals on Earth. They target caterpillars, beetle larvae, and other soft-bodied insects, using venoms, behavioral manipulations, and anatomically precise egg-laying to complete their life cycles. The chalcid wasps, another parasitoid group, contain thousands of additional species. many of which are hyperparasitoids. They parasitize other parasitoid wasps rather than direct hosts. A chalcid wasp may lay its eggs inside a ichneumon wasp larva. That is itself already developing inside a living caterpillar host. Parasitism stacked inside parasitism inside a single living host organism. The ecological impact of parasitoid wasps across insect ecosystems is enormous. They are primary regulators of caterpillar and beetle populations in most forest ecosystems. Without parasitoid pressure, many phytophagous insect populations would increase dramatically, causing significantly greater damage to the plant communities they feed on. The emerald jewel wasp is a small and specialized member of this ecologically enormous group. What makes it stand out within the parasitoid world is not that it uses a living host, but how precisely and specifically it controls that host's behavior. Write in the comments what detail from this story struck you most. Because when researchers first described the antenna-leading behavior in systematic detail in the 1940s, it was considered so unusual that some were skeptical of the reports until it was directly filmed. A wasp leading a living cockroach into an underground hole by grasping its antenna is the kind of thing that sounds wrong even after you have seen it happen. There is one more thing worth saying about what the jewel wasp actually represents that connects this specific insect to questions well beyond entomology. The jewel wasp's manipulation of the cockroach's behavior requires the wasp to have an extraordinarily precise functional model of the cockroach's nervous system built into her own genetically encoded instinctive behavioral programs. She knows DA in a functional sense encoded in her genes, where the subesophageal ganglion is. She knows how to locate it tactily by probing with the tip of her stinger. She knows approximately how much venom to inject and in precisely what sequence. She knows to wait the specific amount of time for the first sting's effect before delivering the second. She knows to wait through the full grooming episode before attempting the leading walk. She knows to grasp the antenna rather than any other structure on the cockroach's body. None of this functional knowledge was learned or observed or transmitted from a parent. All of it was encoded across evolutionary time through the selective survival of wasps, whose programs were more precisely calibrated against the cockroach's specific neuroanatomy. This means evolution has effectively reverse-engineered the cockroach's nervous system and encoded the results into the jewel wasp's behavioral genome. The wasp carries somewhere inside her own 3-centimeter nervous system a functional model of how to disable a specific circuit in another animal's brain, without having any cognitive understanding that she is doing this. This is one of the more philosophically interesting examples of what evolution can produce, not intelligence in the sense of flexible adaptive reasoning about novel problems, but something that looks like extraordinarily precise knowledge, embedded instead in automatic, precisely calibrated behavioral programs, running without awareness or intention in a 3-centimeter emerald green wasp, hunting cockroaches in the leaf litter and undergrowth of tropical forests, doing something that took human neuroscientists decades to partially understand, and getting it right every single time from the first attempt. I want to return to the cockroach in the burrow one final time, not to the detailed biology of what is actively happening to it at that moment, but to what the situation looks like if you could simply observe it from outside. A cockroach sitting motionless in a small sealed underground chamber, slowly cleaning its antennae and body surfaces in a quiet repeating motion, sealed inside a burrow by debris placed there by a 3-centimeter wasp, with a single white wasp egg glued to the underside of its front leg, going nowhere at all and making no attempt to change that situation, not because it lacks the physical capability to move or to leave, but because the specific neurochemical instruction that would send it somewhere was interrupted 4 weeks ago by a precisely targeted injection, and the circuit has never recovered or restarted since that injection. It is not suffering in any way that current science can confirm or measure. It is not aware of its situation in any way that current neuroscience can establish. It is simply present in that chamber doing what the venom left it doing, grooming itself slowly and repeatedly in the dark of the sealed burrow, waiting in a sense for something that is already physically attached to its own leg, and that will eventually consume it from the inside out over the following weeks. The new adult jewel wasp will emerge from that same burrow in approximately 4 weeks, metallic green and fully formed and immediately ready to begin hunting. and go find another American cockroach in the undergrowth nearby, and perform the entire sequence again from the beginning without hesitation, because her genes contain the complete instructions for how to do it. Having never needed to learn a single step of the process from any source other than her own genes,